Welcome to today's Explainer. Right now, we're diving into a massive, and I mean massive, paradigm shift in how we test artificial intelligence in the life sciences. We're talking about OpenAI's brand new GeneBench-Pro. If you've been following AI news lately, you probably know we've kind of hit a wall, right? It's been incredibly hard to trust these models with highly complex, super ambiguous biological data. Well, this new release, it might just be the sledgehammer that finally breaks through that wall. So here's the game plan for today. First, we'll unpack the research taste problem. Second, we'll actually meet GeneBench-Pro. Third, we'll break down its 129 problems across 10 domains. Fourth, we'll look at the evaluation workflow itself. And finally, we'll wrap up with a concrete action plan for developers.
All right, Section 1, the research taste problem, moving beyond guesswork.
To really understand why GeneBench-Pro matters so much, we have to look at the huge bottleneck that's been holding back AI-driven life sciences. Up until now, standard benchmarks have mostly evaluated models on, basic stuff. Pattern recognition, code generation, simple factual recall. It's essentially like giving the AI an open-book test that rewards it for just acting like a static lookup tool, just guessing the expected answer. But here's the thing, real-world computational biology just doesn't work like that. Biology actually demands an AI that can handle messy, ambiguous data, iterate on its own reasoning, and ultimately make really consequential diagnostic decisions.
And this brings us to this absolutely brilliant concept highlighted in the source material, research taste. Research taste is basically defined as those crucial chains of judgment calls that shape an entire analysis. Think about what a real human researcher does, right? They're constantly deciding which questions their data actually supports. They know exactly when early diagnostics should prompt a total change to their model, and they realize when an initial plan just needs to be thrown out and revised. This kind of adaptability is exactly what outdated testing methods have completely failed to measure.
Section 2. Meet GeneBench-Pro, the new standard. Let's look at the timeline here. So before 2026, AI in this space was largely tested on these clean, perfectly synthetic datasets and static Q&A prompts, not very realistic. But then, on June 30, 2026, OpenAI dropped GeneBench-Pro. And as developers quickly pointed out just a couple of days later, this research-level benchmark explicitly drags AI away from those multiple-choice shortcuts. It literally forces the AI to navigate real-world biological ambiguity and test its true end-to-end analytical reasoning. So, the crucial point is how it actually pulls this off. GeneBench-Pro relies on exactly 129 carefully constructed, highly judgment-heavy problems. There are absolutely no shortcuts here. The entire benchmark is designed to measure the full analytical chain.
Section 3, 129 problems, 10 domains, mapping the benchmark.
What's really fascinating here is that the heaviest focus is placed directly on clinical workflows and foundational genetics. We've got a whopping 26 problems dedicated entirely to clinical, pharmacogenomics, and diagnostics. This is huge because it directly addresses the immediate, real-world need for reliable AI and diagnostic workflows. Right behind that, we have population genetics with 21 problems and statistical genetics with 17, which tackle those really foundational analytical challenges.
But, it doesn't stop there. The rest of the problems are spread out across a total of 10 domains and 21 subdomains. We've got 17 problems in quantitative genetics, 17 in regulatory omics, and then it moves down through cancer genomics with 10, proteomics with 7, all the way to niche but vital areas like forensic and microbial genomics. This incredible spread ensures the AI is robust across the whole shebang, from the moment data is generated all the way to complex biological interpretation.
Moving on to Section 4, the evaluation workflow, mimicking real science.
Okay, so having the right problems is honestly only half the battle. How do you actually score an AI's research taste across all these insanely complex domains? Well, GeneBench-Pro uses a very constrained, 4-step, open-ended workflow that perfectly mirrors how an actual computational biologist works in the lab. First up, the input. The model gets messy, real-world datasets complete with missing metadata and noise. Second, the context. It gets just a few brief experimental notes defining the biological question. Third, the target. It's given a specific estimate, which just means the exact target value we're trying to estimate, like a diagnostic threshold. And finally, the process. The model can't just guess here. It has to actively explore the data, validate its assumptions, do iterative checks, and then, and only then, output a justified answer.
The source material has a quote that just brilliantly illustrates the core philosophy of GeneBench-Pro. It says, "The evaluation does not rely solely on matching the final answer. It scrutinizes the reasoning chain, testing whether models can adjust their approach when initial assumptions conflict with the data." This is so important. We're no longer just grading the final output, right? We are demanding that the AI prove its adaptability under pressure. If the data completely contradicts the model's initial hypothesis, the model has to catch that and revise its approach, exactly like a human scientist would.
Section 5. Action Plan for Developers Deploying with GeneBench-Pro.
So, what do we actually do with all this? If you're a developer or a researcher building agents for scientific workflows, you need to pay attention, because this benchmark is designed for programmatic execution. You absolutely have to follow a rigorous setup checklist. First, pin your model versions. Results can degrade super fast as models update, so make sure you record the exact version, temperature, and top P parameters. Second, standardize your evaluation environment. Make sure your GPU memory and context windows can actually handle these massive, messy genomic datasets. Third, track the reasoning, not just the final answers. You need to log the intermediate steps and even the approaches the AI rejected. Fourth, integrate these problems into your CI/CD pipelines for automated regression testing. That way you catch any drift in the model's research taste before you deploy. And lastly, build pre-processing guards into your toolchain to explicitly handle the intentional data noise, like malformed inputs.
If you want to put your own models to the test, and honestly, if you're in this field, you absolutely should, you can find the full evaluation methodology, the problem sets, and all the updates to the scoring rubrics directly at OpenAI's official announcement page. Those 129 problems cover edge cases that standard benchmarks just completely miss, so running the full suite is highly recommended to verify that cross-domain robustness.
As we transition into this totally new era of end-to-end analytical reasoning in computational biology, the stakes are just way too high for fragile, guessing game models. It's time to take a hard look at the tools you're building or using and ask yourself this: is the AI you rely on truly possessing the research taste required for the messy realities of biology, or is it just acting as a static lookup tool? GeneBench-Pro is finally here to help you find out.